So I mentioned ketone bodies a moment ago, and we've seen that they provide fuel and energy for certain tissues. And I'm sorry I did reorder these slides at the last moment, so you might be scrambling. That won't happen the next time around. Most of the acetyl-CoA that's produced by the oxidation of fatty acids in the liver enters the citric acid cycle. However, the remainder is converted to three other metabolites known as ketone bodies. And we're seeing the three, well, no, we're only seeing two here. Acetoacetate is one of those. Beta hydroxybutyrate is another. Not shown here is acetone. But those three metabolites are then acetone, acetoacetate, and beta hydroxybutyrate. This process is known as ketogenesis. I'm sure everyone's heard of the ketogenic diets or ketogenic diet, and there are a number of them. These diets are high in fat, they provide adequate protein, but are very low in carbs, and actually have been utilized, and its first use, that is, was to treat difficult to control epilepsy in children. And what this diet does is it forces the body to burn fats rather than carbs, so that the brain is then using that beta hydroxybutyrate to provide energy. So again, the liver converts fat to fatty acids and ketone bodies. The ketone bodies then replace glucose in the brain as an energy source. Ketosis, which of course is the elevated level of ketone bodies in the blood, leads to a reduction in the frequency of epileptic seizures. Quite interesting. I think one of the first ketogenic diets that became popular for weight loss was perhaps the Atkins diet. And there are a number of others that have been evolved since then. Whether they're good for you or not is not clear. Obviously, for the treatment of epilepsy, at least in children, and there may be some benefit to adults as well, it has a therapeutic effect. So basically, pros and cons I've heard or read about. Well, it does promote quick weight loss. It is suggested that weight loss is not maintained. So this slide shows us the possible fates of glucose 6-phosphate. So this is not covered in the three attachments I have also included in this week, but I want you to see these interrelationships as well. So glucose 6-phosphate has 3 or 4, if you look at acetyl-CoA, that could be included. 3 fates. It can be converted to glycogen to be stored until we need it, but keep in mind as well that we do have limited stores of glycogen. We only store about a day's worth of energy as glycogen. Glucose 6-phosphate can also enter the pentose phosphate pathway and be utilized to produce NADPH and pentose phosphates. Of course, it can also go into the blood as blood glucose, and it can be fed into glycolysis, which is where most of that glucose ends up, to convert pyruvate and acetyl-CoA. Acetyl-CoA, here are actually 3 of the fates that are represented in one of the handout I did attach. It could enter citric acid cycle, it could be converted to cholesterol, or utilized for fatty acid synthesis. And this all occurs in the liver. This table just summarizes some or most of the organs that we have looked at. We didn't look at, of course, the adipose tissue, but organizes the energy reservoirs that are available to this tissue, the preferred substrate or energy source, and if there are any energy sources exported, they are listed here. And there are a few of those. Skeletal muscle we've already looked at, can convert pyruvate to lactate, which can be sent to the liver to produce glucose. And the liver, of course, can use fatty acids, glucose, and or export fatty acids, glucose, and ketone bodies. So we're going to take a look at an enzyme that has a large impact on controlling metabolism or energy production and consumption in mammals. You can think of this enzyme or protein kinase. It is called AMP activated protein kinase, or AMP. It can also, well, it's activated by cyclic AMP, and we'll look at that momentarily as well. But this kinase is responsible for phosphorylating other enzymes that control catabolic and anabolic and/or anabolic processes. So if we have high levels of ATP, this kinase is inhibited. So ATP is an allosteric inhibitor. If we have high levels of AMP, AMPK is allosterically activated and phosphorylates many targets involved in energy production and consumption. So if we have low energy, which of course corresponds to increased concentrations of AMP, AMPK is activated. And in turn, it turns on catabolic pathways. So it positively regulates fatty acid uptake, oxidation, glucose uptake, glycolysis. It also controls our food intake. It also, in muscle, promotes fatty acid uptake, oxidation, as well as glucose uptake, and mitochondrial biogenesis. So muscle tissue is capable of producing or de novo producing mitochondria. On the negative side, we want to turn off anything that consumes energy because we are at a low energy state. So it would inhibit fatty acid synthesis, cholesterol synthesis, gluconeogenesis that occurs in the liver, also fatty acid synthesis in adipose tissue, and inhibit the secretion of insulin. So in effect, activation of AMPK, or AMP-activated protein kinase, turns on catabolic pathways leading to ATP synthesis and turns off pathways that consume ATP. Now we're going to look at the Cori cycle, and this cycle is named for Carl and Gertie Cori. And just a side note, Gertie was the first American female to receive a Nobel Prize. So she's a special lady. We've seen that under strenuous exercise, we create anaerobic conditions in muscle tissue, and pyruvate is converted to lactate. Lactate is then transported through the blood to the liver. So this is what's occurring in the muscle tissue. In the liver, that lactate can be reoxidized by liver lactate dehydrogenase to pyruvate, which of course, to pyruvate at the cost of 6 ATP molecules, can be converted to glucose via gluconeogenesis. And that glucose then can enter the bloodstream and return to that exercising muscle. So this is another cycle, excuse me, called the Cahill cycle, excuse me, or glucose-alanine cycle. And this cycle occurs during extended periods of fasting or prolonged starvation. And what happens is skeletal muscle is degraded as an alternative source of energy, thereby initiating this process. And the skeletal muscle protein degradation, so here we have amino acids from that degradation, are deaminated. We produce alanine in the muscle tissue, which is carried by the blood to the liver, where it can be converted to pyruvate, which enters gluconeogenesis to produce glucose, which can then return to that muscle. That alanine is deaminated, and the ammonium ion will enter the urea cycle. So, glucose-alanine cycle, or shuffle, it's sometimes called. This slide is just another way of looking at the interaction of several, or interconnections of several organs, just in a different way. Included here is the kidney, which we have not looked at before. But the kidney can utilize glutamine to end up being converted, or to essentially end up being converted to glucose. So glutamine can be deaminated to alpha-ketoglutarate and then converted to glucose, which can, of course, be transported through the blood to other tissues. We have the liver, excuse me, that can convert lactate to glucose, utilize amino acids as well, with that ammonium ion going to the urea cycle, which we just saw. And of course, sorry, this is the fed state on this side, fed fasted state here. Responsible then for also creating those triacylglycerols or fatty acids. Here we're using glycogen to glucose, amino acids to glucose. Of course, this is in the fasted state. So lots of interconnections between these three tissues and others. So here's a up-close look at just one of the roles of that protein kinase A, and this is also known as cyclic AMP-dependent protein kinase, which is actually the AMPK that we looked at before. So here we have two hormones, glucagon, epinephrine. We've looked at these before. Glucagon, of course, is responsible for glucose homeostasis, and epinephrine is excreted and controls the fight or flight response. So both of these are going to promote the production of metabolites that can be utilized for energy. So cyclic AMP is called a second messenger. So when glucagon or epinephrine binds a cellular receptor, it triggers a signaling cascade, and that's what this is called. Cyclic AMP is called a second messenger, and its role is to activate protein kinase A, or the cyclic AMP-dependent protein kinase. This plays a key role in the regulation of glycogen, carbohydrate, and lipid metabolism. So protein kinase A, in turn, activates phosphorylase kinase. And this kinase, when it phosphorylates, different enzymes either inhibits them or activates them. And in this case, we want to promote the release of metabolites that can be converted to energy. So we would want to turn off glycogen synthesis. We don't want to store glycogen. We want to release it. So glycogen phosphorylase is activated, and we can then convert that glycogen to glucose. And hormone-sensitive lipase, which plays a role in lipolysis, activates that process, and fatty acids are then released from adipose tissue. And here's a closer look at that glycogen synthase and glycogen phosphorylase activity. Here we have the kinase activity, and we have not talked about phosphatases. At least I don't think so. Anyway, they are responsible for dephosphorylating, excuse me, in this case, and that effect can have different outcomes. As we have seen, kinases, when they phosphorylate, can either activate or inhibit another enzyme. And phosphatases, when they dephosphorylate, can either inactivate or activate. So in the case of glycogen synthase A, it is more active in the dephosphorylated state. It requires the kinase and ATP to be phosphorylated, and it becomes less active. So phosphorylation inhibits glycogen synthase, as we saw in the previous slide. Glycogen phosphorylase is less active in its dephosphorylated state and more active in its phosphorylated state. So one cannot assume that phosphorylation leads to activation. And finally, this slide or diagram is just a summary that allows or organizes, I should say, the location of the processes we have looked at. You don't need to be concerned about the transporters in the mitochondrial membrane, but please remember that the citric acid cycle, electron transport, beta oxidation, ketogenesis, amino acid synthesis and degradation, urea cycle, all occur in the matrix of the mitochondria. The cytosol houses the enzymes for glycolysis, gluconeogenesis, pentose phosphate pathway, fatty acid synthesis, nucleotide synthesis, amino acid synthesis and degradation, and the urea cycle. So next we are going to start the 4th unit, and that will be an introduction to the molecular biology portion of this course.